Welcome to the deep dive. Today, we're diving headfirst into a topic that, well, in one way or another, touches nearly all of us. Cancer. It's an incredibly formidable opponent. I mean, just look at the numbers. It's the second leading cause of death in the Western world, right behind heart disease. And it's startling to consider that almost half of all men and about 1 in 3 women in the United States are projected to develop some form of cancer in their lifetimes. These are truly staggering figures, aren't they? Really highlights cancer's pervasive presence in modern medicine. But here's the question that, often sparks incredible curiosity. What if our own bodies, in their infinite wisdom, already possess a sophisticated defense system specifically built to combat this relentless adversary? Sounds almost like science fiction, doesn't it? Yet it's a complicated, dynamic relationship, a constant, silent biological chess match playing out within us every single day.
And that's precisely the immunological paradox we face with cancer. At its heart, a cancer cell isn't a foreign invader like a virus or a bacterium. It's an altered cell.
Altered self. Okay.
And this fundamental characteristic makes it an incredibly tricky target for our immune system. Think about it. The immune system has this delicate, intricate job. It must be precise, capable of identifying and eliminating these rogue cells without mistakenly launching an attack on our healthy, normal tissues.
Right, that misdirected attack. That's autoimmunity.
Exactly. That's what we call autoimmunity. And the remarkable part is that one of the routine maintenance roles of our immune system is indeed surveillance for cancer cells. It's always on patrol, looking for trouble.
Mm-hmm.
The challenging part, however, is that cancers are, by nature, masters of avoidance. They've evolved astonishingly cunning ways to escape this very recognition and thrive under pressure.
And that's precisely the mission of this deep dive for you, our curious listener. We're going to peel back the layers of this intricate, high-stakes battle. We'll get into, in detail, how our immune system recognizes and fights cancer, how cancer, in turn, fights back and evades detection, and then the truly revolutionary new therapies designed to tip the scales back in our favor. Our goal isn't just to present facts, but to provide you with the nuanced, professional-level understanding of one of biology's most critical and rapidly evolving frontiers, really giving you the insights to master this information like the elite professionals in the field. So let's begin by getting to know the enemy a little better. #tag understanding cancer, the enemy within. Okay, let's start with the basics then, defining what we're actually up against. When we talk about uncontrolled cell growth, you'll often hear the term neoplasm. What is that exactly?
Right. A neoplasm simply refers to an abnormal mass of tissue, essentially a cluster of cells that exhibit unregulated expansion. They're growing when they shouldn't be.
Got it. And there are different types.
Yes, and it's critical to clearly differentiate between two main types of these growths: benign and malignant tumors.
Okay.
A benign tumor is essentially a growth that has a limited capacity for expansion. It reaches a certain size and then usually stops. Crucially, it does not invade the surrounding healthy tissue. It's usually self-contained, almost like a well-behaved guest, you could say.
Right, it stays put.
Exactly.
Yeah.
Malignant tumors, on the other hand, are what we refer to as true cancer. These tumors continue to grow relentlessly, becoming progressively more invasive, pushing into and destroying adjacent healthy tissues. And what truly elevates the danger of malignant tumors is their ability to metastasize.
Metastasis, that's the spread, right?
That's the one. It's this insidious process where small clusters of cancerous cells dislodge from the original or primary tumor. They find their way into the blood or lymphatic vessels, essentially hitching a ride, and are then carried to distant sites in the body. Once there, they establish new colonies, continuing to proliferate and forming what we call secondary tumors. You can almost picture it. A single altered cell gradually transforming, forming a localized benign growth, then becoming a destructive malignant force, and finally spreading its tendrils throughout the entire body.
That spread, that ability to metastasize,
Yeah.
it's undeniably what makes cancer so dangerous. Now, when we classify cancers, I understand it's often based on where they started, the embryonic origin of the tissue.
That's a major way we categorize them, yes.
So the most common type,
The carcinomas.
by far.
Oh,
They
yeah.
make up a staggering 80% to 90% of all cancers. These are tumors that develop from epithelial origins.
Epithelial, like linings. Exactly. Think of the cells that line surfaces, like your skin, the inner lining of your gut, or the epithelial lining of internal organs and glands. So familiar examples like skin, colon, breast, prostate, and lung cancers. Those are all types of carcinomas.
Okay, 80-90%? That's huge. What about the others? You mentioned sarcomas.
Right, sarcomas. These are much less frequent, only accounting for about 1% of cancers, maybe a bit more. They derive from mesodermal connective tissues.
Connective tissues, like bone and cartilage.
Precisely. Tissues that provide structure and support bone, fat, cartilage. So you'll hear about soft tissue sarcomas or osteosarcomas, which are bone cancers.
Got it. And then there are blood cancers.
Yes, a distinct and quite significant category, making up approximately 9% of all malignancies. These are cancers derived from blood cells. This group is a bit more nuanced because they arise at different developmental stages of hematopoietic stem cell differentiation, the cells that make all our blood.
So different stages mean different types of blood cancer.
Exactly. Within these blood cell cancers, we generally talk about leukemias, lymphomas, and myelomas. Leukemias originate from cells that are still in their very early, often undifferentiated stages of development within the bone marrow. They're broadly categorized as either myelogenous or lymphocytic, depending on which lineage of blood cells they affect, myeloid or lymphoid.
And I've heard of acute versus chronic leukemia. What's the difference there?
That's a clinical classification based on progression. Acute leukemia appears suddenly and progresses very rapidly, demanding immediate intervention. Think fast and aggressive. Chronic leukemia, in contrast, is much less aggressive, developing slowly with often mild symptoms over a longer period.
Is the prognosis different?
It used to be quite stark, but what's encouraging, especially with modern treatments, is that many acute leukemias now actually have a good prognosis. Permanent remission is often possible, which really wasn't the case just a few decades ago.
That's hopeful. So that's leukemia. What about lymphomas and myelomas?
Right. Lymphomas and myelomas arise at later stages of blood cell development, typically after these progenitor cells have migrated out of the bone marrow. Lymphomas tend to spread through the lymphatic system, proliferating in secondary lymphoid organs like lymph nodes. That's why you often see them presenting as, swollen lymph nodes.
Makes sense. And myelomas?
Myelomas stem from the uncontrolled proliferation of fully differentiated B cells, specifically plasma cells. Those are the antibody factories. These cancerous plasma cells produce an abnormal mutated form of antibody, often called M protein, which we can detect in the blood and use as a diagnostic marker. Myelomas typically migrate to the bone marrow where they reside, leading to common and often debilitating symptoms like bone pain and anemia.
And multiple myeloma, does that just mean it's in multiple places?
Exactly. The term multiple myeloma refers to the fact that most affected individuals will present with multiple bone lesions, indicating these cancerous plasma cells have taken up residence in several different locations within the bones.
Wow. It really is a disease with many, many faces, depending on where it starts. So, for a normal, well-behaved cell, what actually happens for it to become malignant? How does that transformation occur?
Yeah, that's the core question. Much of our fundamental understanding of this transformation comes from meticulous in vitro studies in lab dishes using primary cells. These are cells isolated directly from a host that, in their normal state, have a limited lifespan and controlled growth.
Okay.
What researchers discovered was remarkable. When these normal cultured cells were treated with specific chemical or physical agents, what we call carcinogens, things like formaldehyde, benzene, asbestos, or even exposure to ionizing radiation, or even certain viruses, their morphology, their shape, and their growth properties could be profoundly altered.
Changed how?
Well, in some cases, this process led to unregulated growth. And strikingly, if these altered cells were then injected into animals, they could actually form tumors. This entire process is termed transformation, or malignant transformation.
So you can essentially make a cell cancerous in the lab?
Under the right conditions, yes. And what's truly fascinating is how these in vitro transformed cells begin to exhibit properties that strikingly mimic actual cancer cells in vivo inside the body. For instance, they show a drastically decreased need for survival factors, like growth factors and serum, which healthy cells are critically dependent on.
They become more independent.
Right. They also lose their anchorage dependence, meaning they don't need to be attached to a solid surface to grow, and they lose their cell contact dependence. Healthy cells stop growing when they touch each other, but these transformed cells just pile up on top of each other. We call that density-independent growth.
Like they ignore the normal rules.
Exactly. And crucially, they also acquire the ability to avoid apoptotic signals, the programmed cell death commands that normally eliminate damaged or unwanted cells.
They refuse to die.
Precisely. And just like cancer cells, these transformed cells can be subcultured indefinitely. They become, in essence, immortal in the lab dish. Studying this in vitro transformation has provided a powerful foundational model for understanding the complex journey of cancer induction.
So it's clearly not just a spontaneous event, it's a series of accumulating changes. The research really highlights this, doesn't it? Emphasizing that cancer isn't an overnight phenomenon, it's more like a confluence of factors.
Absolutely. Our bodies are constantly exposed to various carcinogens, but thankfully our sophisticated DNA repair mechanisms are usually quite effective at preventing cancer from developing. It takes more than just a single hit, usually, to overwhelm those defenses.
So multiple hits are needed.
Generally, yes. And one of the most critical ways this confluence of factors manifests is through chromosomal translocations.
Translocations. That's when bits of chromosomes swap places.
Exactly. Parts of one chromosome break off and then reattach to a different chromosome. This alters the expression and regulation of certain genes located near the breakpoints. We see this frequently in B and T cell leukemias and lymphomas, where these translocations often involve immunoglobulin or T cell receptor gene loci regions of our DNA that are normally highly transcriptionally active in those cell types.
So putting a growth gene next to a very active region.
That's often the case. A prime textbook example that beautifully illustrates this mechanism accounts for 75% of Burkitt's lymphoma cases. It's the translocation of the MYCAT gene from its normal position on chromosome 8 to the immunoglobulin heavy chain enhancer region on chromosome 14. In other Burkitt's cases, the light chain genes might translocate near MYCAT on chromosome 8, causing a similar effect.
And MYCAT is important for growth.
Very. The profound result of these translocations is a significant increase in the synthesis of the MYC protein itself, which is a powerful transcription factor. MYCAT controls many genes that are intimately involved in cell growth and proliferation. So this abnormal surge in MYCAT protein directly leads to unregulated cellular growth. It provides a clear example of how a seemingly small genetic rearrangement can have a profound and devastating impact on cell behavior, turning it cancerous.
That's a perfect illustration of how chaos can truly emerge from subtle genetic changes. And this brings us to the core genetic architects of cancer, the very genes that normally control cell proliferation and survival. Healthy tissues, as you alluded to, maintain a delicate state of homeostasis, right? A balance.
A very tightly controlled balance, yes, between cell proliferation, new cell growth, and programmed cell death, or apoptosis. Any significant imbalance here, whether it's too much proliferation or too little cell death, can really set the stage for tumor development.
So it's about disrupting that balance.
Precisely. The genes involved in these homeostatic processes produce proteins that either encourage or discourage cellular proliferation and survival. Not surprisingly, it's the disruption of these very same growth-regulating genes that accounts for most, if not all, forms of cancer. These disruptions can occur at virtually any point in the cell cycle pathway. From the initial signaling events at the cell surface, through the complex intracellular signal transduction processes, and all the way to the nuclear events that control gene expression.
So many places it can go wrong.
Indeed. For simplicity, though, we can categorize these normal cellular genes, which become implicated in cancer when something goes wrong, into three major groups based on their normal activities.
Okay, what are they?
The first category is oncogenes. These are essentially mutated or dysregulated forms of what we call proto-oncogenes.
Proto-oncogenes, the normal versions.
Exactly. A proto-oncogene is a normal cellular gene whose protein products are involved in promoting cell growth and proliferation. Think of it like a gas pedal for cell growth. When a proto-oncogene undergoes a mutation or becomes dysregulated, it transforms into an oncogene, and its altered expression then leads to uncontrolled cell proliferation, which is cancer. It's like that gas pedal getting stuck in the on position.
Can you give some examples?
Sure. Concrete examples include genes that encode growth factors, such as cousacesis, or genes for growth factor receptors like ERB for the epidermal growth factor, EGF receptor, or CINU, whose overexpression is strongly linked to aggressive breast cancers. Then there are signal transducers, a prime example being the RAS gene family.
RAS, I've heard of that one.
It's a big one. Single-point mutations in RAS have been detected in carcinomas of the bladder, colon, and lung, and overactivity of the RAS oncogene is seen in a striking 30% of all human cancers and almost 90% of pancreatic cancers.
90%, wow.
It's crucial. Even transcription factors like micrike, which we just discussed, Jun and FOS fall into this category. And as we mentioned, viral integration, like what happens with the avian leukosis virus inserting into c-Myc, or the human papillomavirus, HPV, linked to cervical cancer, can also transform proto-oncogenes into their cancerous oncogene counterparts.
Okay, so oncogenes are the gas pedal stuck on. What's the next category?
The second critical category is tumor-suppressor genes, sometimes called anti-oncogenes. These are the brakes on cell proliferation.
The brakes. Makes sense.
In their normal, healthy state, they actively prevent cells from progressing through the cell cycle inappropriately, stopping unchecked growth. Unlike oncogenes, which become problematic when their activity is enhanced, tumor suppressor genes become involved in cancer when they fail to function, when the brakes fail.
So you need to lose the brakes.
Exactly. And a crucial point here is that they typically require a two-hit disabling sequence. For cancer to develop, both copies of the gene, one from each parent, must be lost or inactivated.
Why both?
Because one functional allele, one good copy, is usually sufficient to suppress tumor growth. You need both brakes to fail completely.
Ah, okay. Examples of these.
The TP53 gene, which encodes the p53 protein, is arguably the most critical and frequently mutated genetic abnormality in human cancer. It's found in 60% of all tumors.
60%. What does P53 do?
P53 is a remarkable nuclear phosphoprotein with multiple critical roles. It can induce growth arrest, initiate DNA repair, and if the damage is too severe, even trigger apoptosis programmed cell death. It's a major guardian of the genome. Its prevalence is particularly high in small cell, lung, breast, and colon cancers. Another classic example is the RB, directly linked to hereditary retinoblastoma, a type of eye cancer.
Okay, gas pedals and brakes. What's the third category?
Finally, we have apoptosis genes, which, as the name suggests, control programmed cell death.
The self-destruct mechanism.
Right. These genes can either promote or inhibit apoptosis. Pro-apoptotic genes, like BANCs, act similarly to tumor suppressors. In their normal state, they encourage cell death when something is wrong, thereby inhibiting cell survival. Conversely, anti-apoptotic genes, like BCL2, behave more like oncogenes, actively promoting cell survival even when cells should be dying.
So problems here can also lead to cancer.
Absolutely. A failure of pro-apoptotic genes or an overactivity of anti-apoptotic genes can powerfully encourage the neoplastic transformation of cells and allow them to persist. BCL2, for instance, was originally discovered due to its association with B-cell follicular lymphoma. It plays a vital role in regulating cell survival during normal hematopoiesis and in the maturation of our B and T cells. And interestingly, the Epstein-Barr virus, which causes mono, actually contains a gene with significant homology to BCL2, suggesting it too can suppress apoptosis in infected cells, contributing to its oncogenic potential.
So it's a truly complex interplay. Gas pedals getting stuck on, brakes failing, and the cell's natural self-destruct mechanism being disarmed. To sum up this incredibly intricate section, the research reinforces a fundamental concept. Cancer-causing agents, or carcinogens, and random events that damage DNA, including some viral infections and chromosomal translocations, can accumulate into a condition that allows unregulated cell proliferation and cellular transformation. It's rarely one big event, is it? But rather dangerous series of accumulating hits over time.
That's the key takeaway: accumulation over time.
And this accumulation of hits brings us to the multi-step journey of malignancy. It's clear that the full-blown cancerous state doesn't simply happen overnight.
That's absolutely right. The development of a cancerous state is a gradual, often insidious process. It arises from a series of small, incremental genetic changes, each one nudging the cell closer to uncontrolled replication. This is aptly described as a multi-step process of clonal evolution, driven by successive somatic mutations. Mutations in body cells, not germ cells.
And you differentiate between types of mutations, driver versus passenger.
Yes. We now differentiate between driver mutations, which are those early, pivotal genetic changes that really initiate this cascade, and passenger mutations, which sort of come along for the ride but can also contribute to the overall malignant phenotype later on. Clinical investigations suggest that as few as 2, perhaps as many as 8 key genes can serve as original drivers, with a long list of possible passenger mutations accumulating over time. These collective genetic changes progressively convert the cell from being under normal growth control to a malignant state where all the natural checkpoints have been circumvented.
And the research breaks down this journey into distinct phases, doesn't it? Initiation, promotion.
Yes. Initiation, promotion, progression, and finally metastasis.
Okay, walk us through this. Initiation.
Initiation involves those very first genomic changes or mutations that subtly alter a cell's proliferation potential. By themselves, these initial changes don't necessarily lead to full-blown malignant transformation. It's the first step.
Then promotion.
Promotion is where pre-neoplastic cells that are now precancerous, gradually begin to accumulate and expand. At this stage, the tumor size is generally small. And crucially, the cells are still amenable to our body's natural repair mechanisms. These first two stages, initiation and promotion, can last for very long periods, sometimes decades, and the cells remain susceptible to immune-mediated detection and even chemopreventative agents, meaning there's still a significant potential to reverse the disease's course at this point.
So there's a window of opportunity there. What about progression?
Progression, unfortunately, tends to move much more quickly and aggressively. Genetic alterations occurring during this phase allow for rampant, unchecked cell proliferation. The cells acquire new, dangerous mutations in potential cancer-promoting genes, truly exacerbating this cycle. As you might expect, tumor sizes can grow rapidly in this stage, often becoming clinically detectable.
And then the final stage, metastasis.
Right, the most dangerous stage, metastasis. This is when one or more of these rapidly dividing cells acquire additional mutations that enable them to lose adhesion with their neighboring cells. They no longer exhibit contact inhibition. They gain the ability to invade nearby tissue, allowing them to move outside the original site and even enter blood or lymphatic vessels, where they can then spread throughout the entire body.
Is there a good example of this whole process?
Yes, human colon cancer is a classic example that beautifully illustrates this natural progression. It typically develops through a series of well-defined morphological stages. It often begins as small, benign tumors called adenomas in the colorectal epithelium. These precancerous tumors gradually grow, displaying increasing intracellular disorganization until they acquire the fully malignant phenotype, or carcinoma.
And you can link specific gene changes to these stages.
Remarkably, yes. The scientific literature visually correlates these morphological stages with a specific sequence of gene changes. You see the inactivation or loss of three key tumor-suppressor genes, APC, DCC, and TP53, and concurrently the activation of the KRAS oncogene, which is intrinsically linked to cellular proliferation. This stepwise accumulation of genetic changes, building one upon the other, truly serves as a blueprint for the development of many different cancers. It's not just random chaos.
And this is where it really comes together, isn't it? This concept of accumulated DNA alterations leading to a common endpoint, that's the basis for the hallmarks of cancer.
Exactly. Back in 2000, Hanahan and Weinberg proposed a set of 6 defining characteristics that distinguish a cancer cell. Then, a decade later, in 2011, they updated this list to include 4 more overarching and linked conditions. It's truly an order-within-chaos concept, showing how diverse genetic changes can converge on a common set of capabilities that cancer cells need to survive and thrive.
What were the original 6 hallmarks about?
The original 6 revolved around 3 essential cellular processes. How a cell decides its fate, like growth signals, how it maintains its genetic integrity, or fails to, and how it ensures its own survival, like avoiding apoptosis.
And the 4 later additions, what do they add?
They provide crucial context. These include genome instability, meaning the cancer cells accumulate mutations at an accelerated rate, fueling further evolution. Also, altered metabolic pathways, as cancer cells often reprogram their metabolism, how they use energy to efficiently support their rapid growth and division. Then there's chronic inflammation, which, paradoxically, as we'll discuss more later, can actually help the tumor. And crucially for our discussion today, immune avoidance patterns, the sophisticated ways cancer cells evade detection and destruction by our immune system.
So these hallmarks really show how cancer isn't just about uncontrolled growth, is it?
Not at all. It's a complex constellation of acquired capabilities that allow these cells to survive and thrive against all odds, including the very defenses of our immune system.
And there's another layer of complexity, cancer stem cells.
Yes, adding another layer to this landscape is the concept of cancer stem cells. Growing evidence suggests that within a heterogeneous tumor, a tumor made of different cell types, a specialized subset of cells, the cancer stem cells, are the real engines of tumor growth. They possess unlimited regenerative potential and are the major persistent source of new cells that feed the tumor's expansion.
So they keep the tumor going.
Essentially, yes. The vast majority of the tumor, what we call non-stem cells, each undergo unique mutations and differentiate in novel ways. These non-stem cells often serve as primary immune targets, almost like decoys. They rapidly mutate and express an ever-evolving set of new protein markers, offering potential targets for the immune response. However, there's surprisingly little long-term risk to the tumor if these non-stem cells are recognized and destroyed.
Because the stem cells just make more.
Exactly. Their undifferentiated stem cell parents remain as a constant source of replacement, ensuring the tumor's continued survival and growth. It's a truly sophisticated strategy for long-term survival.
It's a truly sophisticated strategy, a masterclass in adaptation. So to summarize this complex first part, the core idea is that cellular transformation occurs as the result of multiple gene mutations that accumulate in several genes over time and gradually subvert the normal checks on cell growth and survival. And underpinning this, malignant cells display alterations in key cellular processes and microenvironmental conditions. Cell fate decisions, genome maintenance, cell survival, genetic instability, metabolic changes, and immune response patterns. This profound understanding of cancer's intrinsic nature really sets the stage beautifully for understanding the immune system's intricate and often surprising role. Hashtag the immune system's double-edged sword in cancer. Okay, now that we have a clearer picture of the enemy, let's turn to our body's defense system. Despite being cells that originated from our own body neoplastic cells can produce specific tumor antigens that our immune system can, in fact, detect.
That's right.
And most of these tumor antigens are presented on MHC class I molecules. Why is that important?
That's absolutely critical because MHC class I presentation is the primary way that cells signal to cytotoxic T lymphocytes, our killer T cells, that something is wrong inside the cell.
So it's how they flag themselves for destruction.
Essentially, yes. And the scientific literature outlines 4 main ways these tumor antigens arise. First, you have antigens encoded by genes that are exclusively expressed by tumors. These are truly unique to the cancer, not found in normal cells. Second, there are variant forms of normal genes that have been altered by mutation, leading to new, slightly different protein bits, or epitopes, that can be recognized as foreign. Third, you find antigens normally expressed only at certain stages of development, like during fetal life, but which mysteriously reappear in cancer cells in adults. And finally, there are antigens that are simply overexpressed in particular tumors, reaching levels far beyond what you'd find in healthy cells, making them stand out.
Okay, 4 sources, and these get grouped together for therapy purposes.
Broadly, yes, into 2 key categories with significant implications for therapy. First, we have tumor-specific antigens, or TSAs.
TSAs, specific, so they are unique.
Exactly. These are unique proteins, often resulting from DNA mutations that generate completely neoantigens, which are essentially brand new, foreign-like peptides the immune system has never encountered before, or they can be viral antigens from cancer-causing viruses. Because these are truly foreign or new to the immune system, they make ideal immune targets with very little risk of collateral damage to healthy tissues.
Can you give an example of a viral TSA?
Sure. A powerful example can be seen with the Epstein-Barr virus nuclear antigen expressed in Burkitt's lymphoma cells, or the HPV E6 and E7 proteins, which are found in over 80% of invasive cervical cancers. These viral proteins are consistently present in the tumor cells, making them highly recognizable as foreign by the immune system, offering a clear target for intervention.
And this is where it gets truly fascinating. This understanding of virally induced TSAs led directly to the development of the groundbreaking HPV vaccine, right?
Absolutely. It's rightly celebrated as the first clinically approved cancer vaccine. It's truly one of the closest things we have to a cancer cure, in a prophylactic sense, preventing the cancer from ever developing.
And cervical cancer is a huge global problem.
Tragically, yes. It's the 3rd leading cause of death among women globally, responsible for approximately 275,000 deaths annually. And about 80% of these occur in developing countries where regular screening like pap smears isn't widely available.
And HPV itself is incredibly common.
The human papillomavirus, yes. It's the most common sexually transmitted infection globally. It's implicated in over 99% of cervical cancers, but its reach extends far beyond that. It's linked to most cases of vaginal, vulval, anal, penile, and oropharyngeal, that's head and neck cancers, as well as genital warts.
Are specific types worse than others?
Yes. Of the hundreds of HPV genotypes, types 16 and 18 are responsible for over 70% of cervical cancers, while types 6 and 11 are primarily associated with genital warts.
And the vaccines target these.
Exactly. The HPV vaccines like Gardasil and Cervarix target these specific high-risk types and are remarkably effective, between 95% and 99% effective at preventing HPV infection.
So why aren't vaccination rates higher?
That's the challenge. Societal adoption. Despite their proven effectiveness, vaccination rates remain surprisingly low in many regions. Barriers include costs, especially in developing countries, and significant social and ethical concerns, along with widespread misinformation. Many parents, for instance, view HPV vaccination as an issue for the future, assuming their children aren't sexually active.
But that's often not the case.
Unfortunately, no. CDC data shows nearly half of high school students in the U.S. have had sexual intercourse, with many starting before age 13. The vaccine regimen is most effective when completed prior to exposure and produces the most robust immune response in 11 to 12-year-olds.
So timing is critical.
Absolutely. This really highlights the critical importance of physician recommendations to overcome these barriers and ensure vaccination prior to sexual activity for the most robust and long-lasting immune response.
Okay, so TSAs are ideal unique targets. What's the other category? TAAs.
Right. Tumor-associated antigens, or TAAs. These are more common, but also more challenging for the immune system.
Why challenging?
In contrast to TSAs, TA is not unique to neoplastic cells. Instead, they are normal cellular proteins that display unique or abnormal expression patterns. For example, they might be expressed only during fetal development or at extremely low, almost undetectable levels in healthy adults, but become highly induced or dramatically upregulated in tumor cells.
Ah, so they're self-proteins, just expressed weirdly.
Exactly. And because they are normocellular proteins, they are subject to our natural self-tolerance mechanisms, making them harder for the immune system to target without potentially inducing autoimmunity against healthy tissues that might express low levels.
Can you give some examples of TAAs?
Sure. A classic subset is oncofetal tumor antigens. These are TAAs that normally appear only early in embryonic development, before the immune system is fully competent and establishes robust tolerance to them. When these fetal proteins mysteriously reappear on neoplastic cells in an adult, they can be recognized as aberrant and potentially induce an immune response.
Like AFP and CEA?
Precisely. Two well-studied examples are alpha-fetoprotein, or AFP, and carcinoembryonic antigen, CEA. AFP, the most abundant fetal protein, is typically found at very low levels in healthy adults, but significantly elevated levels are seen in liver, ovarian, and testicular cancers. Similarly, CEA is an oncofetal membrane glycoprotein found in fetuses, and increased levels are seen in about 90% of advanced colorectal cancer patients.
Can you diagnose cancer with these?
Not definitively on their own, as trace amounts can be found in some healthy adults or other non-cancerous conditions. But they are incredibly useful for monitoring prognosis and treatment efficacy. For instance, a rise in CEA after colorectal carcinoma removal is a strong indication that tumor growth may have resumed, signaling a need for further intervention.
So they're good for tracking. Any other TAA examples?
Yes, another type includes products of overexpressed oncogenes. For instance, some tumor cells express the epidermal growth factor, or EGF receptor, at levels 100 times greater than normal cells. Melanoma cells, for example, can express melanotransferin, P97, at truly massive levels, hundreds of thousands of molecules per cell, compared to maybe 8,000 in normal cells.
And we can target this overexpression.
Yes. Targeting such overexpressed products has led to significant clinical successes. A prime example is Herceptin, a humanized monoclonal antibody that targets HER2, an EGF-like receptor, which is overexpressed in about 25–30% of metastatic breast cancers. This powerfully demonstrates how even normal self-proteins, when aberrantly expressed in these high numbers, can become viable therapeutic targets.
Okay, so let's reiterate that crucial distinction then. Tumor-specific antigens are non-self proteins that arise from DNA mutation or cancer-causing viruses, yielding peptides recognized by the immune system as foreign, unique targets.
Right.
Whereas cancer-associated antigens are normal cellular proteins that display abnormal expression patterns and thus are not foreign. However, in some instances, these can still be recognized by the immune system or strategically used clinically to monitor or therapeutically target the cancer, more complex targets.
That sums it up perfectly.
This brings us to the dynamic dance, immunoediting. You mentioned earlier there was debate about whether the immune system even played a role in cancer.
For over a century, yes, there was a heated debate. Some argued that because cancer cells arise from self, they would be protected by tolerance mechanisms, essentially flying under the radar. But decades of rigorous data from both animal models and clinical studies have now definitively shown that the immune system plays a clear, vital, and incredibly complex role in both tumor cell identification and eradication.
Even before the immune system gets involved, the body has other defenses, right?
Absolutely. It's worth noting that even before the immune system fully steps in, our bodies have multiple intrinsic and extrinsic mechanisms designed to prevent cancer. Intrinsically, within the cell, you have pathways like the nucleotide excision repair, or NER pathway. These can induce cell senescence, a permanent stop to growth, or even apoptosis, programmed cell death, at the first signs of trouble, and they actively facilitate DNA repair. If those fail, extrinsic mechanisms involve environmental signals instructing cells to activate internal pathways, leading to growth arrest or apoptosis. For example, if epithelial cells lose their normal connections to the surrounding matrix due to malignant changes, that disruption itself can trigger death signals. It's a multi-layered safety net.
But if those initial nets are breached, the immune system steps in. How does it control cancer?
There are three proposed mechanisms. The first two are broadly familiar, actively destroying viruses known to transform cells, like HPV or EBV, and rapidly eliminating pathogens and regulating inflammation to prevent chronic pro-cancerous environments. But the third, immunosurveillance, is particularly relevant here.
Immunosurveillance, the immune system actively looking for cancer cells.
Exactly. The idea that our immune system continually monitors for and destroys newly transformed cells, which, as we said, is no small feat, given they are self cells.
And there's evidence for this.
Significant, compelling evidence. For instance, immune-suppressed individuals, like AIDS patients or transplant recipients on powerful immunosuppressive drugs, have a significantly higher incidence of several types of cancer. Animal models similarly show that almost any form of immunosuppression leads to increased incidence of both spontaneous and induced cancers. These observations powerfully demonstrate the potential of the immune system to hold cancer at bay.
But here's where it gets really interesting. The immune system might not always be helpful. It can sometimes promote tumors.
That's the twist. While the immune system clearly inhibits tumors, recent data also points to potential pro-tumor influences, shifting our understanding to a much more nuanced view. This led to the immunoediting hypothesis, which acknowledges both tumor-inhibiting and tumor-enhancing processes.
How did that idea come about?
A key discovery in the mid-1990s, using RAG2 knockout mice that entirely lack adaptive immunity, T and B cells, was particularly surprising. When tumors were induced in both these RAG2 knockout mice and normal wild-type mice, and then cells from these tumors were transferred to healthy, immunocompetent recipients, a significant portion, up to 40% of the tumors that originally grew in the immune-deficient RAG2 knockout mice, were actually rejected by the healthy recipients.
Rejected, so they were easier for a normal immune system to see.
Exactly. This strongly suggested that tumors growing in an immune-deficient environment are more immunogenic, meaning they're easier for the immune system to recognize than those arising in an immunocompetent environment. This groundbreaking insight led to the powerful idea that the immune system doesn't just eliminate, it actively sculpts or edits tumors through a Darwinian process of selection.
Survival of the fittest, or sneakiest cancer cells.
Precisely. Those cancer cells that manage to survive immune winnowing are simply better able to outwit the immune response, gaining a crucial survival advantage in this biological arms race.
And this immunoediting happens in phases.
Yes, this profound process can be divided into three sequential phases. The first phase is elimination, which aligns with the traditional view of immunosurveillance. The immune system effectively identifies and destroys newly formed cancer cells. However, under this intense selective pressure, some cancer cells can acquire mutations that allow them to resist immune destruction.
Okay, so some survive elimination. Then what? Equilibrium.
Exactly. The second phase is equilibrium. This is a fascinating state of balance where low levels of abnormal cells persist, but their proliferation and spread are held in check by the adaptive immune response. Clinical evidence suggests this phase can last for decades, whether the tumor is actively treated or not. Environmental and lifestyle choices can significantly influence its duration, making it a potentially fruitful window for therapeutic intervention.
Held in check for decades.
Wow.
But eventually they can escape.
That's the third and final phase, escape. This is when the most aggressive and least immunogenic of the residual tumor cells, those that have truly won the immunoediting battle, begin to thrive and spread. This happens often, ironically, with help from certain immune pathways themselves. During this phase, immune activity can shift from being anti-tumor to actually being pro-tumor, creating a permissive environment for cancer growth.
The immune system starts helping the cancer.
In some ways, yes. This critical shift explains why some cancers, initially held in check for years or even decades, eventually break through and become clinically aggressive, often in a form that is much harder for the immune system to tackle.
So it's a dynamic, evolving, and sometimes brutally efficient relationship. To crystallize this, a core concept is immunoediting is a model that describes how the developing tumor-specific immune response identifies and kills cells, elimination, or immune surveillance, maintains a balance between death and survival of cancer cells, equilibrium, and critically also fosters the selection and survival of cells that develop immunoevasive mutations, escape.
That captures the essence of it perfectly.
This dynamic takes us to the immune players and their messages, detailing the innate and adaptive pathways involved in this complex battle. Our sources highlight that there are, surprisingly, both good anti-tumor and bad pro-tumor forms of inflammation in the response to cancer. That sounds like a double-edged sword.
It absolutely is. On the good, or anti-tumor side, we see innate responses powerfully dominated by immune-activating M1 macrophages, highly efficient cross-presenting dendritic cells, and of course natural killer or NK cells. These cells and the specific cytokines they produce, like IFN gamma, help elicit strong T helper 1, Th1, and cytotoxic T lymphocyte CTL responses. These are generally associated with a good prognosis and impressive tumor regression.
Okay, so M1 macrophages, DCs, NK cells, Th1, CTLs, those are the good guys here. What about the bad inflammation?
Conversely, in tumors that are more likely to progress and metastasize, the immune cell infiltrates often include anti-inflammatory M2 macrophages and myeloid-derived suppressor cells, or NDSCs. Adaptive responses in these cases are often skewed towards the Th2 pathway and sometimes involve Th17 or Treg cells. All of these, unfortunately, are associated with poorer clinical outcomes and reduced survival times.
So M2 macrophages, MDSCs, Th2, Th17, Tregs, they can hinder the fight. Let's delve into the innate inhibitors of cancer first. NK cells seem important.
NK cells, or natural killer cells, were among the very first cell types recognized for their inherent ability to directly destroy tumor cells. Their name itself derives from this innate killing capacity. Scientific studies show that mice deficient in NK cells exhibit an increased incidence of lymphomas and sarcomas. And in humans with genetic defects specifically affecting NK cells like Chiari-Higashi syndrome, there's an associated increase in certain cancers. It underscores their critical importance in initial tumor surveillance.
How do they recognize the cancer cells if they're self?
Their recognition is truly fascinating in its simplicity and effectiveness. They use a series of surface receptors that respond to a combination of activating and inhibiting signals. One key mechanism is recognizing missing self.
Missing self.
Yes. Many transforming viruses and some cancer cells try to hide from T cells by reducing the expression of MHC class I molecules on their surface. Since NK cells typically recognize cells with normal MHC levels as self and thus leave them alone, a distinct lack of MHC class I molecules signals an abnormality, allowing NK cells to identify and kill these missing self tumor cells.
Clever. What about other signals?
Likewise, tumor cells undergoing stress or DNA damage can express altered-induced self signals like specific molecules called NKG2D ligands. These powerfully engage NK cell activating receptors.
And once they recognize a target.
Once engaged, NK cells unleash cytolytic granules containing perforin and granzymes to directly kill target tumor cells, poke holes and inject toxins, essentially. Indirectly, they also contribute significantly by secreting IFNO, a potent anticancer cytokine that enhances MHC expression on dendritic cells, further stimulating strong CTL responses down the line.
So they kill directly and help activate other cells. What about macrophages?
Beyond NK cells, activated macrophages also play a significant role. They're often seen clustering intensely around tumors. And the presence of pro-inflammatory M1 type macrophages in these infiltrates strongly correlates with tumor regression and better outcomes.
How do macrophages kill cancer cells?
Like NK cells, they express SCC receptors on their surface, enabling them to mediate a crucial process called antibody-dependent cell-mediated cytotoxicity, or ADCC. They destroy cells coated with antibodies. Their anti-tumor activity also involves potent lytic enzymes, the production of reactive oxygen and nitrogen intermediates that can damage cancer cells, and the secretion of tumor necrosis factor alpha, TNF-I, which has well-documented potent anti-tumor activity.
And eosinophils, too. I thought they were just for allergies and parasites.
Surprisingly, yes. Eosinophils have a recently discovered intriguing role in cancer immunity. Studies in mice have shown that those lacking eosinophil chemoattractants like eotaxin-1 or stimulatory cytokines like IL-5 were found to be more susceptible to carcinogen-induced cancers, while mice with higher eosinophil levels were more resistant. This points to a previously unsuspected yet potentially important layer of innate immunity against certain cancers.
Fascinating. Okay, that's the innate side. Let's transition to the adaptive cellular processes and the cells involved. T cells are key here, obviously.
Absolutely critical. In animal studies, it's unequivocally clear that animals lacking alpha-beta T cells or even gamma-delta T cells are more susceptible to various induced and spontaneous tumors. It powerfully highlights the importance of T cells in cancer defense.
And specifically CTLs, the killer T cells.
CTLs, or cytotoxic T lymphocytes, specifically CD8 plus T cells, are considered absolutely crucial in direct tumor killing. They've recognized tumor antigens presented on MHC class I molecules on neoplastic cells. Strong anti-tumor CTL activity correlates significantly with tumor remission. They are particularly credited with maintaining that critical equilibrium phase of cancer immunoediting, holding the cancer in check for potentially long periods.
So vaccine strategies often target CTLs.
Naturally, yes. This understanding has driven the development of many cancer vaccines aimed at inducing strong anti-tumor CTL responses. And as we know, CD8 plus T cells rarely work alone. They need help from activated dendritic cells to get primed, and Th1 type helper cells to foster their full, robust response.
Is there more direct evidence for adaptive immunity's role?
Yes. Further compelling evidence comes from studies where mice treated with low-dose carcinogens but not yet developing cancer suddenly erupted with full-blown cancer when their adaptive immune system was specifically knocked out. Importantly, blocking NK cell responses didn't cause this eruption, but specifically blocking CD8 plus T cell responses, or the key cytokine IFN, did.
Wow, so adaptive immunity is actively suppressing those early cancers.
It profoundly underscores the critical role of adaptive immunity not just in eliminating, but in sculpting tumors, actively selecting for less immunogenic cells over time. And one cunning way tumors achieve this immune evasion is by increasing their expression of PD-L1, a molecule which binds to the PD-1 receptor on CTLs, effectively applying a brake and inhibiting their engagement, preventing destruction.
We hear a lot about TILs in cancer therapy research, tumor-infiltrating lymphocytes. What's the story there?
Right. In clinical studies, the frequency of tumor-infiltrating lymphocytes, or TILs, which are a diverse mix of T cells, NKT cells, and NK cells found directly within the tumor mass, strongly correlates with cancer regression. For instance, in one ovarian cancer study, a remarkable 38% of women with high TIL numbers survived more than 5 years, compared to just 4.5% of those with low TILs. That's a huge difference.
Does the type of TIL matter?
Absolutely. Beyond just the sheer numbers, the type of infiltrating cells matters even more prognostically. A high frequency of tumor-specific CD8 plus T cells, or an elevated ratio of CTLs to Treg cells, is consistently associated with enhanced survival and better patient outcomes.
So having lots of killer cells and fewer suppressor cells is good.
Precisely. Conversely, the specific depletion or targeted disruption of Treg cells or regulatory T cells has led to dramatic cancer regression in several mouse models. This is now a major focus of ongoing clinical studies in humans trying to block those Tregs.
What about B cells and antibodies? Do they help?
The role of B cells and antibodies in cancer immunity is a bit more complex and, at times, even contradictory. On one hand, B cells can generate anti-tumor antibodies that foster tumor cell recognition and lysis. For example, these antibodies can bind to tumor cells, making them targets for ADCC by NK cells and macrophages, which have Fc receptors that bind to the antibodies, so they can flag cells for destruction.
But here's the twist in the plot, isn't there? Sometimes antibodies can actually be detrimental.
Yes, and it's a surprising one. Some anti-tumor antibodies, which we sometimes call enhancing antibodies or historically serum blocking factors, can actually play a detrimental role.
How?
Instead of protecting, they can bind to tumor-specific antigens and literally masks them from the cytotoxic T lymphocytes, or CTLs, preventing the CTLs from recognizing and attacking the cancer. They hide the target. Or they can form antibody-antigen complexes that inhibit ADCC by binding to FCC receptors on immune cells like NK cells or macrophages, effectively disarming these immune effectors.
So they can block the killers or block the helpers?
Right. This complex, sometimes counterproductive role of B cells in cancer immunity is why their precise contribution is often seen as less straightforward and less obviously beneficial than that of T cells in many contexts.
Okay, that's complicated. Finally, let's discuss the role of cytokines in cancer immunity. These are the signaling molecules, the messages.
Yes, these powerful signaling molecules are the language of the immune system, directing and orchestrating responses. Animal models where specific cytokine pathways are eliminated have helped us identify the critical role of individual cytokines. Generally, cytokines associated with the Th1 pathway and strong CTL responses are consistently linked to cancer elimination.
Like IFN gamma.
IFN, interferon gamma, both type I, alpha beta, and type 2 gamma interferons, is clearly important. It can exert direct anti-tumor effects on transformed cells. Critically, it powerfully enhances MHC class I expression on cancer cells, making them much better targets for CD8 plus T cell recognition and destruction.
What about IL-12?
IL-12 has also received significant attention for its remarkable ability to enhance anti-tumor immunity. Administration of exogenous IL-12 can protect mice from chemically induced tumors, and IL-12 deficient mice develop more papillomas, underscoring its protective role. This is largely because IL-12 strongly drives Th1 and CTL responses, creating a robust anti-tumor microenvironment. It's a key component in some promising current cancer trials.
And TNF-alpha, tumor necrosis factor, sounds like it should be good.
TNF was actually named for its direct anti-cancer activity because when injected into tumor-bearing animals, it induces hemorrhage and necrosis tissue death of the tumor itself. However, its role in the human body is surprisingly complex and dual-natured. Depending on the context and the specific type of cancer, TNF can have both tumor-inhibiting and, paradoxically, tumor-promoting effects.
So it can help or hurt?
Can, making its therapeutic application a nuanced challenge. It's a powerful molecule that can be either friend or foe, depending on the situation.
It's clear there are a lot of players on different teams, sometimes even switching sides. So, to synthesize this intricate section, several non-adaptive cells and processes are involved in cancer recognition and removal, including NK cells, M1-type macrophages, and possibly eosinophils, along with the crucial cytokines produced by these cells, such as IFN, TNF-A, and IL-5, respectively.
Correct.
And on the adaptive side, anti-tumor CTLs are the adaptive cell type most associated with cancer eradication, although activated DCs and Th1 type cells likely also assist powerfully in this pathway.
Yes.
We also learned that anti-tumor antibodies can bind to the surface of cancer cells, allowing cells with Fc receptors, such as NK cells and macrophages, to induce ADCC in tumor targets, though sometimes they hinder.
Right, that caveat is important.
Finally, the cytokines most consistently connected with anti-tumor responses include both type I and type II interferons, TNF-A, and IL-12, all of which are associated with strong Th1 cell and CTL responses, pivotal for fighting cancer.
A good summary of the key players and messages. #tag when immunity goes wrong, promoting cancer.
Now for what might be the most counterintuitive part of this relationship. We've talked about how the immune system fights cancer, but ironically, some elements of the immune response can actually promote cancer survival and progression.
It seems paradoxical, doesn't it?
It really does. It truly underscores that the immune response is a delicate balance, and sometimes those vital breaks fail or are simply misdirected, leading to unintended consequences.
Exactly. A prime example is chronic inflammation. While acute short-term inflammation can be a beneficial part of healing prolonged, chronic inflammation is widely believed to create a pro-tumor microenvironment.
How does chronic inflammation help cancer?
Through several distinct mechanisms. Firstly, sustained inflammatory responses increase cellular stress signals and can lead to genotoxic stress, producing highly damaging reactive oxygen species, or ROS. This, in turn, significantly increases mutation rates in nearby cells, fostering the very tumor genesis we want to avoid. It creates instability. Secondly, leukocytes, the white blood cells involved in chronic inflammation, secrete various growth factors and cytokines that can directly induce proliferation in non-immune tumor cells. They essentially feed the cancer's growth.
So immune cells can accidentally help cancer grow.
In this context, yes, it's a truly unwelcome paradox. And thirdly, inflammation is pro-angiogenic and pro-lymphangiogenic.
Meaning it helps grow blood vessels.
Exactly. It actively increases the growth of local blood and lymphatic vessels. This supplies oxygen and nutrients to solid tumors, literally feeding their rapid expansion. It also aids tumor cell invasion into surrounding tissues and transport through newly constructed lymphatic vessels, both of which are critical hallmarks of aggressive cancer and metastasis. It helps them spread.
Wow. So chronic inflammation is bad news for cancer control. What about those tumor-enhancing antibodies you mentioned?
Yes, another counterintuitive finding. There was this surprising historical observation that immunizations with anti-tumor antibodies, rather than protecting against tumors as one might expect, sometimes actually enhanced tumor growth. It flew in the face of conventional wisdom.
How could that happen?
This perplexing phenomenon was seen in early studies where immune cinera from animals with progressive tumor growth were found to block the CTL lysis of target cells in vitro, whereas serum from animals with progressive tumors did not. In 1969, a pioneering discovery by Ingegerd and Carl Erik Hellstrom further demonstrated this. They showed that children with progressive neuroblastoma had remarkably high levels of a blocking factor in their serum, while those with regressive neuroblastoma did not.
Blocking factors, what are they?
Since then, similar serum blocking factors have been associated with a number of human tumors. In some cases, the anti-tumor antibody itself acts as a blocking factor, presumably by simply binding to tumor-specific antigens and physically masking them from CTLs, making the cancer cells invisible again. However, in many cases, the blocking factors are not antibodies alone, but rather antibody-antigen complexes, clumps of antibodies stuck to antigen. These complexes have been shown to directly block CTL responses, and they may also inhibit ADCC by binding to FDC receptors on NK cells or macrophages, effectively disarming these immune effectors.
So they interfere in multiple ways.
Yes. This is a key reason why most clinical antibodies used today to treat solid tumors are designed very carefully to avoid directly targeting tumor-specific antigens in a way that could lead to this blocking effect. It's a known hazard.
Okay, so chronic inflammation and blocking antibodies. What else hinders the immune response?
Then there's the pervasive issue of immunosuppression in the tumor microenvironment, the area immediately around the tumor.
The tumor's neighborhood.
Exactly. Tumor cells themselves, and even certain immune cells that infiltrate a tumor, can secrete soluble factors that create a locally immunosuppressive environment, effectively putting out the immune system's fire right where it's needed most.
What kind of factors?
We see increased expression of things like TGF-A and indoleamine 2-calibre-3 dioxygenase, or IDO, both powerful inhibitors of Th1 responses in various human cancers. The cytokine IL-10 also plays a duplicitous role, sometimes promoting tumors intolerance, but at other times encouraging anti-cancer innate immune responses depending on the context. It's complicated. Similarly, while TGF-meat might actually inhibit early tumor growth, its presence in later stages of cancer often encourages progression, possibly by blocking local dendritic cell activation and directly inhibiting T-cell function.
And the suppression is just local, right at the tumor.
It appears to be quite localized, yes. Compelling studies have shown that while a primary tumor persists in one part of the body, similar cancer cells introduced to a new site on the same mouse can actually be robustly rejected by the immune system. This indicates the tumor creates its own protective, local immune bubble.
And specific immune cells contribute to this bubble.
Yes, this localized immunosuppression is often driven by specific immunosuppressive cells that actively contribute to this permissive environment. We're talking about regulatory T cells or Treg cells, myeloid-derived suppressor cells or MDSCs, those M2 macrophages we mentioned, and Th2 cells. Their presence redirects immunity away from the effective Th1 pathway, which is crucial for killing cancer, creating a permissive environment that allows tumor evasion and survival.
So to encapsulate these complex, often paradoxical interactions, leukocyte infiltration is important for tumor eradication. However, prolonged chronic inflammation and tumor-enhancing antibodies correlate negatively with survival, actively hindering the immune response. And crucially, the presence of immunosuppressive cells like MDSCs, M2 macrophages, and Treg cells, along with the cytokines they produce, such as IL-10 and TGFO, in the tumor microenvironment fosters tumor survival and immune evasion.
That's the challenge we face. The tumor actively fights back by manipulating the immune system itself.
All of this points to how truly sophisticated and cunning cancer is at surviving. It's a master of disguise, constantly adapting to evade our body's best defenses.
It truly is. Despite the immune system's clear capabilities and the expression of tumor antigens, the sobering fact that so many individuals still die from cancer each year highlights that the immune response is often, tragically, ineffective in the long run. This is largely due to the intense selective pressure applied by the anti-tumor immune response itself, which favors the evolution of escape mutants, those cancer cells that manage to successfully evade the immune response. This, as we discussed earlier, is the very essence of immunoediting.
The cancer cells evolve to hide better.
Exactly. Over time, cancer cells have evolved several common and highly effective evasion pathways.
Let's walk through some of those common evasion pathways, starting with how cancer cells can reduce their visibility to the immune system. You mentioned reduced MHC expression.
Yes. Reduced MHC expression in tumor cells is a major one. Defects in antigen processing and presentation are remarkably common among these escape mutants.
How does that happen?
It can involve mutations that lead to a general reduction in MHC class I expression on the cell surface. Or the cancer cells might just secrete their tumor-specific antigens rather than properly presenting them on MHC. Sometimes there are defects in crucial molecular machinery, like the transporter associated with antigen processing, or TAP-E, or in beta-2 microglobulin. Both of those are absolutely crucial components for proper MHC class I assembly and presentation. Even insensitivity to IFNO, the cytokine that normally powerfully enhances MHC class I expression, can contribute.
And the end result is invisibility.
Pretty much. All of these ultimately result in decreased MHC class I presentation of tumor antigens, profoundly inhibiting CD8 plus T-cell recognition. It's a direct, effective way for the cancer cell to become invisible to our most potent killer T-cells.
But wait, didn't you say NK cells kill cells lacking MHC class I?
Ah, good point. There's a double layer of evasion here. NK cells should recognize missing MHC class I. However, tumors are clever enough to also decrease the ligands, the signals that bind to activating receptors on NK cells.
So they hide from T cells and NK cells.
Exactly. It's like they found a way to be invisible to both key types of cytotoxic immune cells. And tragically, the absence or significant reduction of MHC molecules on tumor cells is generally a strong indication of cancer progression and carries a poor prognosis.
Okay, that's hiding. What about resisting the attack? Resisting apoptosis?
Another major evasion strategy. Tumor cell resistance to apoptotic signals. In other words, cancer cells disabling their own self-destruct mechanisms. As cancer evolves, neoplastic cells increasingly develop profound resistance to programmed cell death, apoptosis. This, of course, creates a severe imbalance between proliferation and cell death, leading to uncontrolled expansion of the tumor cell population.
How do they become resistant?
This resistance can arise from defects in the extrinsic pathway, which involves cell surface death receptors like FAS, TRAIL-R, and the NKG2D receptor. These normally receive external signals telling the cell to die, or it can stem from changes in the intrinsic mitochondrial pathways of apoptosis, the cell's internal self-destruct mechanism. This means either decreases in pro-apoptotic signaling components like members of the BCL2 superfamily, such as BAX, which normally push the cell towards death, or increases in anti-apoptotic players like survivin, both of which powerfully encourage enhanced cell survival and cancer progression.
So they jam the self-destruct signals.
It's a direct override of the body's natural quality control system, allowing flawed cells to persist and multiply.
And finally, a significant evasion tactic involves poor costimulatory signals and immunosuppressive microenvironments. Remind us about costimulation.
Right. We've often discussed the crucial two-signal model for T-cell activation. Signal 1 from the T-cell receptor recognizing a peptide MHC complex, and signal 2, the costimulatory signal, typically provided by molecules like CD80 or CD86 on antigen-presenting cells interacting with CD28 on T-cells.
And you need both signals.
Both are absolutely essential for full, robust T-cell activation and proliferation. Without both, T-cells often enter a state of anergy or functional unresponsiveness.
So how do tumors exploit this?
The inherent problem with tumors is that, as self-somatic cells, they naturally have poor immunogenicity and tend to lack these crucial costimulatory molecules, like CD8086, that are vital for T-cell activation. Without sufficient numbers of professional antigen-presenting cells APCs in the immediate vicinity of a tumor, and with few strong stimulators to drive the activation of these APCs, responding T-cells may receive only signal 1, a partial activating signal.
Leading to anergy, shutting down the T cell.
Exactly. This often leads to clonal anergy, a state of functional inactivation, and immune tolerance, where the T cells simply stop responding to the tumor and become ineffective. Furthermore, T cells that do interact with these poorly co-stimulating tumor cells can actually be induced to act as immunosuppressors themselves, becoming TREG cells. These TREG cells then express powerful immune-inhibiting cytokines and utilize co-modulatory molecules like CTLA-4, all of which act as significant impediments to further effective immune activation against the cancer.
So the tumor not only fails to stimulate properly, it actively creates suppressor cells.
It's a cunning, multi-pronged strategy for the tumor to not just hide, but to actively shut down the immune response around it, ensuring its own survival.
It's a truly formidable opponent indeed. To wrap up this section, transformed cells employ several sophisticated strategies to evade the immune response, including reduced MHC class expression to become invisible, anti-apoptotic responses to disable their self-destruct buttons, and poor or blocked co-stimulation of T-cell responses, which collectively generate a microenvironment of profound immune suppression.
A tough combination to beat.
Understanding these intricate evasion tactics is precisely what informs the next generation of revolutionary therapies. Hashtag harnessing the immune system breakthrough immunotherapies. Okay, now let's turn the page to some incredibly hopeful news. Present-day cancer treatments are increasingly multifaceted. We have the traditional pillars, surgery, radiation, chemotherapies, but also hormonal therapies, targeted therapies, and crucially, a rapidly expanding arsenal of immunotherapies.
Immunotherapies are truly ushering in a new era of cancer treatment, fundamentally changing the game.
How are they different from, say, chemotherapy?
Unlike traditional treatments that directly attack cancer cells, immunotherapies are precisely defined as any treatment designed to specifically revive, initiate, or supplement our in vivo anti-tumor immune responses. The core focus is on activating the immune system itself, creating the right signals, or providing the right cells that will guide the immune system in the optimal direction, allowing the body to take care of the rest.
harnessing the body's own power.
Exactly. This paradigm shift represents a profound leap in our approach to cancer.
And this isn't a completely new idea either. There's fascinating historical context, right? Coley's toxins.
That's right. Back in 1891, a pioneering cancer surgeon named William B. Coley began experimenting. He injected bacteria directly into the inoperable tumor of a patient with bone cancer and observed surprising success. This was long before the advent of chemotherapy or radiation, so clinicians had very few options back then. Coley and others continued this novel form of immunotherapy for years, using various bacteria or bacterial products that became famously known as Coley's toxins.
Was it controversial?
It was. Published reports often met with skepticism due to variable results and were eventually overshadowed by the more predictable, though often toxic, chemo and radiotherapy. But today, we understand that Coley's observations were likely based on sound immunologic reasoning. The immune stimulatory characteristics of his toxins probably powerfully boosted natural anti-cancer immune activity, leading to tumor regression in some of his patients.
So he was onto something, just way ahead of his time.
Absolutely. It was a remarkable foreshadowing of modern approaches, decades ahead of its time.
Okay, let's get into the modern approaches, starting with what many think of as the magic bullet approach. Monoclonal antibodies, or MABs.
Right. These are meticulously designed antibodies produced in the lab that selectively bind to specific tumor-specific or tumor-associated surface markers on cancer cells. They can essentially mark the cancer cells for destruction by our own leukocytes, or sometimes they're designed to deliver a conjugated payload of toxins directly to the cancer cell.
How many are approved now?
We now have over a dozen FDA-approved MABs in clinical use, primarily directed against cell-type-specific surface molecules.
Can you give some key examples?
Sure. Rituximab targets the B-cell marker CD20 and is widely used for non-Hodgkin's lymphoma and chronic lymphocytic leukemia, CLL.
Yeah.
While it does destroy both cancerous and non-cancerous B cells, our immune system can regenerate new ones from hematopoietic stem cells, making this an acceptable and manageable side effect.
Okay. Another big one is Herceptin.
Exactly. Trastuzumab, commonly known as Herceptin, targets HER2, a growth factor receptor that is notoriously overexpressed in about 25-30% of breast and stomach cancers. Herceptin has been incredibly successful in treating HER2 positive breast cancers, dramatically improving patient outcomes.
And there's a truly compelling anecdote involving Ron Levy at Stanford that really illustrates the precision, right? The anti-idiotype antibody.
Yes, a classic story. Back in the day, they treated a man with terminal B-cell lymphoma that had unfortunately metastasized throughout his body. Because it was a B-cell cancer, all the cancerous cells expressed the exact same idiotype, the unique antigen binding part of their surface antibody. Livy's team painstakingly produced a mouse monoclonal antibody that was specific for this patient's unique idiotype, a truly personalized antibody.
And it worked.
When injected, this anti-idiotype antibody selectively bound only to the cancer's B lymphoma cells, making them susceptible to complement or ADCC-mediated lysis. After just 4 injections, the patient's tumors began to shrink dramatically, and he entered an unusually long period of remission.
Amazing, though probably expensive back then.
Incredibly costly and time-consuming, yes. But the fundamental concept of such personalized, precise targeting is very much alive and thriving in cancer research today, become more feasible.
And antibodies can also carry toxins, ADCs.
Building on that precision, yes, we now have antibody-drug conjugates, or ADCs. These are quite literally MABs coupled with potent toxins. It could be radioactive isotopes, powerful chemotherapy drugs, or immunotoxins like diphtheria toxin. The ingenious goal is to deliver these payloads at incredibly high local concentrations directly and selectively to the cancer cells, largely sparing non-malignant cells and minimizing systemic toxicity.
Like a guided missile for chemo.
That's a great analogy. Mylotarg was an early anti-CD33 ADC, but it was later pulled due to poor results and safety concerns. Shows it's not always easy. However, more advanced ADCs are now successfully FDA approved and in widespread use. One targets CD30 for relapsed Hodgkin's lymphoma. Another, called TDM1 or Kadcyla, is an immunotoxin form of Herceptin that targets HER2 plus metastatic breast cancer. It binds precisely to HER2 positive cancer cells and then introduces a toxin called DM1, which blocks cell division. It's chemo delivered right to the target.
So to summarize this section, the key concept here is that monoclonal antibodies, ABs, whether used alone or with cytotoxin conjugates that bind to cell type-specific or overexpressed surface markers, can be strategically used to target these cancer cells for recognition and elimination by the toxin or by our own phagocytic immune cells.
Precisely. leveraging antibody specificity.
Okay, moving from antibodies, we can also engineer the immune cells themselves, give them new instructions.
Yes, this brings us to T cells on a mission, the powerful concept of expanding or even creating tumor-specific T cells designed to hunt down cancer.
You mentioned TILs earlier, tumor-infiltrating lymphocytes. They're naturally there, but often don't work.
Exactly. Early on, researchers observed significant lymphocyte infiltration in solid tumors, which suggested these cells might be a natural source of tumor-specific immunity. Indeed, TILs, a mix of tumor-reactive T cells, can be isolated from patients' blood, lymph nodes, and directly from their tumors. However, despite their presence, cancer often persists, indicating these TILs are often anergic in vivos, functionally inactive, and unable to perform their anti-tumor duties effectively within that suppressive tumor microenvironment.
So the idea is to take them out, wake them up, and put them back.
That's the core idea behind TL expansion therapy, particularly pioneered for metastatic melanoma, a very aggressive skin cancer. The process involves collecting TILs directly from the patient's tumors, then expanding them in vitro in the lab in the presence of high-dose IL-2 to overcome their anergic state and supercharge them. After the patient undergoes lymphodepleting treatments, basically, clearing out some existing lymphocytes to make space and reduce suppression, large numbers of these activated autologous from the patient's own body TILs are infused back.
And does this work?
It has shown significant tumor regression in almost half of the patients treated this way, with approximately 10% experiencing long-lived or complete remission. Truly remarkable for such an aggressive cancer previously considered untreatable.
But outcomes vary.
Yes, outcomes can be disparate, possibly due to the presence of Treg cells mixed in with the TILs, which compete for IL-2 and suppress immune responses. To mitigate this, many such adoptive cell transfers now strategically use peripheral T cells rather than directly isolated TILs, hoping to avoid expanding immunosuppressive cells along with the killer cells.
And these T cells can last a long time.
That's a major advantage of this cell-based therapy over MABs. These autologous T cells can be quite long-lived in the patient, offering remarkably durable responses, sometimes lasting years.
Okay, TILs are one approach, but the really cutting-edge stuff seems to be CAR T cells.
Ah, yes, CAR T cells, or chimeric antigen receptor T cells. This is a fast-moving field and represents a true paradigm shift in immunotherapy.
What's the basic idea, chimeric receptor?
The groundbreaking concept, pioneered by Israeli immunologist Zilig Eshar back in 1989, was to create an artificial T-cell receptor. This chimeric receptor combines the antigen-binding specificity of a B-cell receptor, specifically the antibody variable region.
Which can bind directly to antigens without MHC.
Exactly. It binds directly to native antigens on the cell surface, bypassing the need for MHC presentation, which tumors often downregulate. And this B-cell receptor part is fused to the potent cytotoxic signaling machinery of activated T cells.
So you get the direct recognition of an antibody plus the killing power of a T cell.
Precisely. The idea was to give T cells a brand new powerful way to see cancer and attack it, bypassing the tumor's clever MHC evasion strategies.
And these have evolved over time, first generation, second generation.
Yes, the concept is truly revolutionized through iterations. First generation CARs had a relatively simple design. An extracellular antigen binding region linked just one intracellular signaling domain, usually CD3, which is part of the normal T cell receptor complex. The variable region of the CAR was designed to recognize a known cell type specific antigen, like CD19 on B-cell leukemias. Patient T cells were then genetically modified with the CAR construct, expanded in vitro, and reinfused.
How well did they work?
Unfortunately, early trials showed limited persistence of these T cells in the body and relatively short-lived anti-tumor effects. They didn't stick around or pack enough punch.
So they needed improvement.
Yeah.
Second generation.
This led to the development of second-generation CARs, which crucially added one or more costimulatory signals. This is the equivalent of adding both the critical signal 1, antigen recognition, and signal 2, costimulation, that T cells need for full activation, all in one molecule. These more complex designs, incorporating the antigen binding domain, a intracellular signaling domain like CD3 zeta, and one or more costimulation domains like CD28 or 41BB, all in a single transmembrane receptor, yielded far more promising and sustained results. Second-generation CR T cells persisted longer in patients and demonstrated significant, often dramatic, destruction of malignant B cells, with an astonishing 90% response rates in patients with relapsed or drug-resistant disease.
90%, that's incredible.
A highly significant improvement over standard therapies, offering hope where little existed before, particularly in certain leukemias and lymphomas.
But there were side effects, serious ones.
Yes. With this powerful new therapy came severe side effects. Patients experienced serious neurological symptoms and, alarmingly, a life-threatening systemic inflammatory response called cytokine release syndrome, or CRS.
Cytokine storm.
Essentially, yes.
Mhm.
This intense, body-wide inflammatory response from the massively activated CIR T cells caused high fevers, severe flu-like symptoms, drops in blood pressure, and even profound neurological impacts, tragically leading to some patient deaths in early trials.
Could they manage it?
Fortunately, subsequent trials showed that CRS could be managed and often reversed in most patients with the judicious use of anti-IL-6 monoclonal antibodies, like tocilizumab, allowing these crucial trials to resume safely.
So the field kept moving, third-generation CARs.
Despite these early challenges, the truly miraculous improvements seen in many patients with previously dire prognosis fueled the rapid development of third-generation CARs and beyond, often referred to as having accessories. These updated iterations strategically add one or more advanced features to the second-generation model to enhance their effectiveness and safety.
Like, what kind of accessories?
For instance, T cells redirected for universal cytokine killing, are engineered to express potent anti-tumor cytokines like IL-12, along with their CAR, directly modifying the tumor microenvironment to make it less suppressive. Armored CARs are designed to resist the tumor's immunosuppression by either lacking or directly blocking co-inhibitory molecules like PD-1 within the CAR T cell itself. And for crucial safety, self-destructing CARs are designed with built-in suicide triggers, often activated by a specific drug, allowing them to be eliminated if severe side effects occur. While conditional CARs are engineered to be off until specifically activated by a precise administered drug, adding another layer of control.
That's amazing engineering, but I bet it's expensive.
Of course, all these sophisticated enhancements and personalized approaches come with a cost. The initial sticker shock for CAR T-cell therapy could be up to $800,000 per patient, which sounds astronomical.
It does.
However, it's vital to put this into perspective. It's comparable to the lifetime expense of other potentially curative adoptive cell transfer therapies, like bone marrow transplantation, or even the cumulative cost of repeated cycles of traditional chemotherapy for relapsed cancers. Both CAR T-cell therapy and bone marrow transplantation are considered living drugs because of their remarkable potential for self-renewal and lifelong impact. Since many of the most successful CAR T-cell trials have required only a single injection and resulted in high, durable cure rates, the cost is increasingly viewed in light of its potential for sustained remission and even for many patients, a cure. It profoundly changes the long-term economic and health outlook.
And the results justified the first approvals.
Absolutely. The excitement is warranted. The first FDA approval for a CAR T-cell therapy came in August 2017. Tisagenlecleucel T, marketed as Kymriah. It was approved for use in children and young adults aged 3 to 25 with relapsed or refractory acute lymphoblastic leukemia, ALL. In a multi-center preliminary clinical trial, an astonishing 83% of patients achieved complete remission within the first 3 months of treatment.
83% remission in relapsed ALL.
It's incredible, especially for a patient population with a previously devastating clinical prognosis and very few other treatment options. Crucially, no deaths due to CRS were reported in these pivotal trials, marking a monumental shift in cancer treatment and offering a beacon of hope for many families.
So, to summarize this incredibly exciting and rapidly advancing area, adoptive transfer of autologous T cells, whether with or without modifications like CARs, can be used as a longer-lived cell-based therapy to effectively treat cancer, offering durable and often curative responses.
A powerful new pillar of therapy.
Next, let's talk about therapeutic vaccines, which are designed to train or re-educate the immune response. How are these different from, say, the HPV vaccine?
That's an important distinction to clarify. Most vaccines we think of, like the flu shot or childhood immunizations including HPV, are prophylactic vaccines. They prevent infection or disease before it happens by priming the immune system to recognize future threats. Therapeutic vaccines, however, aim to actively enhance or redirect an existing immune response after infection or exposure to the relevant antigen, or in the case of cancer, after the cancer is already developed.
So the HPV vaccine prevents, but a therapeutic vaccine treats existing cancer.
Exactly. To reinforce this, the HPV vaccine is incredibly effective, up to 99%, at preventing infection with the strains that cause cervical cancer in unvected women. But that same vaccine is critically ineffective in women who are already infected with HPV or who already have established cervical cancer. So designing vaccines for patients with existing cancer-causing infections or established neoplastic cells is a unique challenge. It requires a very different strategy to reroute an already engaged but often insufficient or misdirected immune response, aiming to supercharge it against the existing disease.
How did researchers first approach this?
Early cancer vaccine studies explored methods for enhancing tumor-specific antigen presentation to the immune system. For instance, in laboratory settings, mouse dendritic cells, those crucial antigen-presenting cells, were cultured with GM-CSF, a hyaloid-stimulating growth factor, and fragments of tumor cells. These ex vivo stimulated DCs were then infused back into mice. These reinfused dendritic cells successfully activated both TH cells and CTLs, specifically against the tumor antigens, leading to robust tumor immunity when the mice were later challenged with live tumor cells. It was a proof of concept.
Did this lead to human therapies? Like Prevenge?
Yes, this elegant strategy eventually led to Sipulsil T, or Prevenge, which was the first FDA-approved therapeutic cancer vaccine, specifically for metastatic prostate cancer. This therapy uses autologous patient dendritic cells, which are collected from the patient's blood, and then treated in vitro with a fusion protein. This protein consists of prostatic acid phosphatase, or POP, a tumor-associated antigen commonly found in prostate cancer cells linked to GM-CSF. These stimulated APCs are then reinfused into the patient to stimulate a powerful T-cell response against PAP expressed on the prostate cancer cells.
How well did Prevenge work?
Initial results showed only modest survival increases in the general population, maybe a few months on average, and it came with a steep cost of around $93,000 for 3 infusions, which initially dampened interest.
But there was a recent update.
Yes, a more recent exciting development found significantly better outcomes, specifically in African American men with prostate cancer compared to matched white male counterparts. This is particularly important given that African Americans have historically fared more poorly in prostate cancer prognosis and treatment response. The precise mechanism for this difference is currently under intense investigation, but it highlights the potential for tailored benefits.
Interesting. What about using viruses as vaccines? TVEC.
Another fascinating and more recent approach is TVEC, which is a genetically modified oncolytic herpes virus.
Oncolytic means cancer killing.
Right, and this virus has the GM-CSF gene integrated directly into its viral genome to help stimulate immune cells. When injected directly into melanoma lesions, the virus enters cancer cells, replicates furiously within them, and ultimately causes tumor cell lysis, effectively exploding the cancer cells and releasing tumor antigens. Patients treated with TVEC have shown observed reductions in tumor size at the injection site.
Does it work elsewhere in the body too?
What's truly promising is that when TVEC is combined with checkpoint blockades, which we'll discuss next reductions, we're even seen at distant untreated sites. This strongly suggests that this combination therapy may trigger a systemic immune activity against previously unrecognized tumor-specific antigens throughout the body. The local explosion alerts the whole immune system.
And people are working on vaccines targeting those unique neoantigens.
Yes, beyond these, highly promising research is delving into neoantigens, those truly occult tumor-specific antigens that are unique to each individual specific tumor, arising from random DNA mutations. Because they are truly non-self, they escape the immune system's self-tolerance mechanisms and can be powerful targets. Researchers are developing personalized vaccines using protein lysate, or, more commonly now, messenger RNA, mRNA, collected or sequenced directly from individual patient tumor cells. This personalized information is then used to create a vaccine introduced back into the patient in a context specifically designed to elicit a strong, tailored immune activation against their unique tumor mutations.
Truly personalized medicine. How's that going?
These individualized strategies are showing some very promising results in early trials, though none are yet widely clinically available. It represents a real frontier of personalized cancer treatment.
So the key concept for this section is therapeutic cancer vaccines are precisely designed to redirect or enhance the anti-cancer response and use strategies like infusion of autologous dendritic cells loaded with tumor antigens, in vitro stimulation and expansion of autologous cells, or targeted presentation of hidden tumor neoantigens to the immune system.
Exactly, training the immune system to fight better.
And now, for what many consider one of the most transformative breakthroughs in modern oncology, truly a game changer. Releasing the brakes with checkpoint blockade. We've talked about how aggressive cancers create inhospitable microenvironments and often lack those crucial costimulatory signals. Remember, T-cell receptor engagement without that vital CD28 co-stimulation leads to energy.
Functionally shutting down the T cell, making it unresponsive.
Yes. So instead of trying to press the gas, this approach takes off the brakes.
That was the revolutionary hypothesis. Instead of trying to provide the T cell activation gas, like co-stimulation, that's missing or weak in the tumor environment, what if we instead release the brakes by blocking co-inhibitory molecules that are actively suppressing the immune response? This idea was powerfully supported by groundbreaking studies, particularly by James Allison and his colleagues. They showed that monoclonal antibodies against CTLA-4 could induce dramatic cancer rejection in animals with existing tumors.
CTLA-4 is one of these brakes.
Yes, CTLA-4 is a key co-inhibitory molecule found on T cells that acts like a powerful brake, dampening their immune responses early during activation, mainly in lymph nodes.
And blocking it worked.
Yes. This pioneering work culminated in the landmark FDA approval of ipilimumab, or Yervoy, in 2011. This was the first in a new class of anti-cancer drugs called checkpoint inhibitors. Ipilimumab specifically blocks human CTLA-4, thereby releasing the brakes on the T cells. It initially saw truly significant success in treating malignant melanoma, dramatically improving patient survival in a disease that was previously almost universally fatal. Its use has now expanded to several other cancer types.
And there's another major checkpoint pathway, PD-1.
Right, building on that incredible success, the next generation of checkpoint inhibitors emerged. Monoclonal antibodies specific for PD-1, or programmed cell death protein, and its ligand PD-L1.
How is PD-1 different from CTLA-4?
PD-1, like CTLA-4, is another crucial co-inhibitory molecule found on T cells that dampens their activation, but it primarily functions later within peripheral tissues and the tumor microenvironment itself, rather than during the initial activation phase. Critically, its ligand, PD-L1, is frequently expressed on cancer cells themselves and often indicates a poor prognosis for patients.
So the tumor uses PD-L1 to shut down T cells directly.
Exactly. When PD-1 on a T cell engages with PD-L1 on a tumor cell, or other cells in the microenvironment, it sends a powerful stand-down signal, effectively telling the T cell to stop its attack right there at the tumor site.
And we have drugs targeting this pathway too.
Yes. Multiple FDA-approved drugs now target either PD-1 itself, like pembrolizumab, Keytruda, and nivolumab, Opdivo, or its ligand PD-L1, like atezolizumab, Tecentriq, durvalumab, Imfinzi, and avelumab, Bavencio. These powerful checkpoint blockade strategies effectively release the brakes on the host's T cells, allowing previously suppressed but activated CD8 plus T cells to carry out their destructive programming with less interference from regulatory cells and other immunosuppressive cues from the tumor microenvironment.
So you're unleashing the T cells that are already there but being held back.
That's the core idea, allowing these cells to do what they're designed to do.
But taking off the brakes of the immune system sounds like it could have side effects.
Indeed. There is an unfortunate, but perhaps not unexpected side effect to unleashing this powerful immune response. Generalized dysregulation of the immune response.
Autoimmunity.
Essentially, yes. Because these checkpoint molecules, CTLA-4, PD-1, are also critical for maintaining self-tolerance and preventing autoimmunity throughout the body in healthy individuals, blocking them can lead to acute inflammatory and autoimmune symptoms. These are collectively called immune-related adverse events, or IRAEs. They can affect various organ systems, including the skin, rashes, itching, gastrointestinal tract, colitis, diarrhea, liver, hepatitis, and endocrine glands, thyroiditis, hypophysitis, adrenal insufficiency. Some endocrine effects can be irreversible.
So it requires careful management.
Absolutely. These side effects can be serious and require prompt recognition and management, often with immunosuppressive drugs like steroids. But despite these serious side effects, checkpoint blockade is unequivocally a valuable new tool in cancer treatment and marks a fundamental turning point in the cancer battle. It has produced durable remissions, even cures, in patients with cancers previously thought untreatable, truly highlighting the paramount importance of the immune response in this struggle.
So the key concept here is, one of the most promising new anti-cancer immunotherapies is checkpoint blockade, where monoclonal antibodies recognizing co-inhibitory molecules such as CTLA-4, PD-1, or PD-L1 are used alone or in strategic combination to interrupt the negative regulation of tumor-specific T cells, thereby unleashing their full cytotoxic potential.
A revolutionary approach. #tag outro.
Wow. We've covered a tremendous amount of ground today, haven't we? From delving into the intricate origins and complex mechanisms of cancer,
The genetics, the hallmark.
to exploring its complex, often contradictory relationship with our own immune system, the immunoediting, the evasion tactics,
The double-edged sword aspect.
And finally, diving into the incredible breakthroughs in immunotherapy, the antibodies, the CRTs, the vaccines, the checkpoint inhibitors that are profoundly reshaping the landscape of cancer treatment as we know it.
It's truly been quite a journey through the science. What's most fascinating here, I think, is how our deepened understanding of immune activation and regulation, even its self-tolerance mechanisms and potential for misdirection, has fundamentally reshaped how we approach this disease. The concept of cancer as altered self cells and the inherent challenges that poses for the immune system, along with the dynamic balance of immunoediting, truly underpins all these powerful new strategies. Understanding this helps us master this information like the elite professionals in the field aim to.
And while it's important to remember that there's still no single magic bullet or cure on the immediate horizon for all cancers.
No, unfortunately not yet.
Our expanded knowledge of cancer genomics and immunology, combined with this rapidly evolving and incredibly powerful therapeutic toolbox, truly offers a more promising prognosis for many patients than ever before, even just a decade ago.
Absolutely. It's a testament to relentless scientific inquiry and human ingenuity. The progress has been astounding.
And this journey of discovery naturally raises an important question for all of us to ponder.
Considering the incredible adaptability of cancer cells, right, and their proven ability to evolve new escape mechanisms under immune pressure, what future immune evasion strategies might we expect to see them develop next? And how might our brilliant immunotherapies need to evolve to counter them? It's an ongoing arms race. What stands out to you from today's deep dive thinking about that future?
A truly thought-provoking question to leave you with, and one that encourages continued curiosity. We certainly encourage you to continue your own deep dive, perhaps exploring the references provided in our source material for further knowledge. Until next time on the deep dive, stay curious.